Good morning.
Yeah, please let me know my voice is clear.
Yes, ma'am.
Okay. So we were here, no, in the last class. So we have just discussed about these 5 phases of cell cycle, isn't it?
Yes, ma'am.
Yeah, anyone please? Yes. So if everybody here, we can start, but only 28 peoples. You all had your breakfast?
Yes, ma'am.
Yeah, good. Only there are 31 students are here. So 3 minutes more to 10:15. Tell your friends to join quickly. exam? Your opinion? I know some of you face some difficulties during the quiz exam. Can you hear me?
Yes, ma'am.
Yeah, because I'm not using the earphone today, that's why I'm asking. Okay, what about your quiz exam? Yesterday's one. It's fine?
It's okay.
No any comments? The person 094. Is it okay you to join today? You can unmute and you can talk with me. 094.
Yes, ma'am.
Yeah, is it okay to you join today?
Okay, ma'am.
Okay. So I'm sorry for your message. Right? So please take care of your family.
Okay, ma'am. Thank you.
Right. Shall we start? Because I don't have time. I also have exam duties. Yeah, there are 79 students. So we will start. They will join, I hope. Samina, are you there?
Yes, ma'am.
Yeah, please inform to your batch to join to the same link for the cell cycle theory. Right? So we will continue with the
Recording in
cell cycle,
progress.
right? So before moving to the cell cycle, I need to ask some questions. So I think more than 1 month, no, we discussed these things, right? Tell me, what is chromosome? Okay, one by one. Just I will ask some question to trigger your mind, right? So chromosome, what is that? Please unmute and talk with me because each and every time it's difficult to me to check the chat box. Otherwise, I will check the chat box at the end, right?
It contains a genetic material, basic information, the basic sequence matter.
Okay. Any other answers? What about the structure? Okay, I hope you all have paper and pen with you. So while I'm explaining, please take notes with your own key points, right? So the chromosome is a long continuous piece of DNA which contains the genetic information for a cell, right? Next one. Centriole and centrosome. What's that? Centriole and centrosome. So centriole is a barrel microtubule structure, right? And two centrioles arrange themselves perpendicularly to form a centrosome, right? And another one. What is this? Please have a communication with me. You can unmute and talk. What is this? Centromere. What is centromere?
Two sister chromatids are joined.
Yeah, that. the region. You need to say the region of the sister chromatids that would be joined there, right? So we can say that centromere is constricted region. So you need to say the word region or place, region. So region of the chromosomes that hold the sister chromatids together, right? So we can say if it is sister chromatids, this region. So this region is known as the centromere, right? And what is chromatid? You said chromatid. So chromatid is one strand, right? So chromatid. So you need to remember all the things. It is one strand of the chromosomes. So we can say that is after it has duplicated for the cell division. This one is when this is duplicated or separated. So this is chromatid. One is chromatid. So altogether we can say these two sister chromatids. Right? Now tell me why cell division is important. So we already discussed in the previous classes. Why do cell divide?
During the process of reproduction.
For the reproduction, okay.
And new cell generation.
Cell generation. Okay.
To replace the damaged or old cells.
Yeah, replace the damage or old.
For the cell growth.
Yeah, good. The last one, please repeat. So you said growth. Okay, we can say growth and repair. So repair or replace. So repair or replace of dead cells. And also we can say for the asexual reproduction. Right? So for this, we can simply say this is the main reason for the cell division. So now define the term cell division. Could you please define the term cell division? Or cell cycle. Cell cycle. Please define the term cell cycle.
The sequence of
We can say, yeah, one person still. Yeah, tell me.
the sequence of event takes place from one cell cycle to another.
Okay. Any other answers?
Series of events duplicating DNA and dividing the cytoplasm, resulting to daughter cells.
Yeah. So here you are saying duplication, regeneration, and you are saying some steps, isn't it? So we can say simply, so we can collectively say that process, right? So the cell cycle is the sequence of growth and division of a cell. So you need to say sequence of growth and division, right? So simply you can say there are some sequence of steps happening for the growth and division. So collectively we can say that the cell cycle is the sequence of growth and division of a cell. And that cell cycle, it consists of three major phases, right? So interphase, mitosis, and cytokinesis, right? So, and the interphase will be divided into again three phases, G1, S, G2. So G1 means G1 and G2 are growth phases. So G1 is primary growth phase, G2 is secondary growth phase. So in between that, there is one phase is synthesis phase. So there the DNA will be replicated. So these three stages are collectively known as interphase, right? And the next one is M phase. So that one is mitosis, and next one is final one is cytokinesis, right? So we can say the cell cycle or cell division. Those refer to the series of events that take place in a cell that leading to its maturity and subsequent division, right? So these events includes duplication of its genome and synthesis of the cell organelles, followed by the division of cytoplasm, right? So what about the human cells? Is that a prokaryotic cell or eukaryotic cell? Eukaryotic cells. So human cells exhibit typical eukaryotic cell cycle. So it takes around 24 hours to complete one cycle of growth and division, right? So, and also we can't say all the eukaryotic cells taking this amount of time. So the duration of the cell division, so it may be varies according to the organism to organism. So it varies from organism to organism, and also we can say it varies to cell, right? Sometimes in one organism, the time duration for the cell cycle may be varied to cell also in a particular organism, right? So we will discuss about these phases one by one. So this image is collectively simply explaining that. See here, that first one is G1, that is cells mature, that means growth phase. Then S phase, that is for DNA copy, that means DNA replication. Then G2, that cell prepare for division. So in G2, it's ready to go for the division, right? Then that is followed by the M phase, right? That mitosis phase. Then finally, the cytokinesis happen. Then the results will be the daughter cells, right? So this is the simple explanation for that. Right, one by one we will discuss. Interphase. So interphase also known as the resting phase of the cell cycle. Right, so interphase is the time during which is the cell prepare for division by undergoing both cell growth and replication. So we already told that in the interphase G1 is G2. So G means for growth, S for synthesis. So here both growth and replication of DNA is happening, right? So if we talk about the time duration, so this interphase occupies around 90 to 95% of time. So interphase taking 90 to 95% of time of the overall cycle. Then remaining 5 to 10% will be occupied by the cytokinesis, right? So first we will discuss about the G1 stage, right? So G1 stage is the first growth stage after the cell division. So we already told here, this is the first growth stage after the cell division. See here the daughter cells coming, then again it's starting, right? So this is the first growth stage after cell division, and here the cells mature by making more cytoplasm and organelles. So here first this is a growth stage, and the maturations of cells also happening here by making more cytoplasm and the organelles. And also cells carries on its normal metabolic activities, right? We can say cells carries on normal metabolic activities, right? So here in this image, you can observe these red arrows are indicating the growth of cell. The cell grows in size, right? So again, we will talk about the G1 phase. The G1 phase is the phase of the cell between the mitosis and initiation of replication of the genetic material. So it's in between the, so G1 is in between the mitosis and S, right? And during this phase, the cell is metabolically active. So normally metabolically, this phase is metabolically active and it continues to grow. So here these red arrows are indicating that. So these continue to grow, right? But you need to remember here that it continues to grow without replicate its DNA, right? It's continue to grow without replicates its DNA, right? So without replicating the DNA, the cell grows in size. This is a simple explanation for G1 phase. Okay. Next one. So next one is S phase. So S standard for synthesis stage, right? So here the DNA replication takes place during this phase, right? So DNA copy or DNA replication will be happen. So if the initial quantity, so for example, if the initial quantity of DNA in the cell is denoted as 2N. Then what will be the results after the replication? What will be the results after the replication?
Uncopied DNA form.
Yeah, then replication means copying happening. So the result will be 4N. So however, the number of chromosomes does not vary. Right? So if the number of chromosomes during G1 phase was 2N, it's remain 2N at the end of the phase S. Right? So and here in the image, you can observe some other things in this part. Please check here the centrosome. How many centrosomes are here? 1. Then check here. How many here? Centrosome?
2.
2. So, something happens to the centrosome also. So what happened here? The centrosome also divides into 2 central pairs. Right? So here the centrosomes also will be replicated. Right? So here the chromosomes. So synthesis will be happen. DNA replication will be happen. So after that, it will be have 2 copies. Right? This one original DNA and 2 identical copies of DNA. Right? This is all about the S stage. Then the G2 stage, growth stage 2 or secondary growth stage. Right? So here we can say this is a second growth stage and it occurs after DNA has been copied. Right? So this is occur after DNA copied. Right? And all cell structure needed for division are made. Right? And both organelles and proteins are synthesized. right? So we can say that during this phase, the RNA, protein, and other macromolecules required for the multiplication of cell organelles, because this is the stage which is ready for the cell division, right? So before that, it's all the things need to multiplicate, right? Then it will ready for the cell division. Then spindle will start to form, and cell will growth, and cell growth are produced as the cell prepare to go for the M phase, the mitotic phase, right? Here you can observe. So you can see that pair of centrosomes and the nucleus and nucleolus and chromatin threads. So you can observe here chromatin threads. And here also cell, it grows in size, right? So now the cell is somewhat ready for the, it's altogether it's ready for the cell division. Right. So next one is M phase. Right. So in the M phase, this is the mitotic phase. In this phase, that the phase of the equal division as the cell undergoes complete recognition to give birth to a progeny that has the same number of chromosomes as the parent cells, right? So here we can talk about here the division of the nucleus. Also, it's called as karyokinesis, and only it occurs in eukaryotes, and it has 4 stages, and it doesn't occur in some cells such as brain cells. So what we will discuss this one. What will happen if there is cell cycles happen? The M phases are happen in the brain cells, right? So here you can observe these images and tell me what is mitosis. What do you have idea for that?
It is a cell division which gives rise to identical daughter cells.
Identical, yeah, good. Any other answers? Any other answers? What about that? It is happening in plant cell and animal cells? Is it? Why you all silent? Shamina?
Yes, miss.
Yeah. So what do you have idea for mitosis? No idea? Then tell me, what about the mitosis happening in plant cells and animal cells? Or only plant cells or only in animal cells?
Both.
Both, yeah. So the plant and animal cell divisions occurs as a part of their life cycle, right? So here, the cell division both in plant and animal cells can be divided into 2 types. So we can say that is vegetative cell division and reproductive cell division. So if you talk about the vegetative cell division, so that is which produces the genetically identical 2 daughter cells. So that is known as mitosis. in the reproductive cell division, so which produces 4 gametes, which is containing half of the chromosome numbers, right? So that is not identical, that is half of the chromosome numbers as in a vegetative cell, right? That is referred to as meiosis, right? So now we are talking about the mitosis, right? So this mitosis, is the phase of the cell cycle where the nucleus of the cell is divided into 2 nuclei with an equal amount of genetic material, right? So here we can say that nucleus of the cell divided into 2 nuclei, right? And with an equal amount of genetic materials which is in the both daughter cells, right? So we can say that mitosis is essential for the growth of the cell and the replacement of worn out cells, right? So I can say some other features of the mitosis. In each cycle of the cell division, 2 daughter cells are formed from the parent cell. Right here, these are some features. 2 daughter cells will be formed from the parent cell, and the cell is also known as equational cell division because the chromosome number in the parent cell and daughter cell is the same, right? And in plants, mitosis leads to growth of vegetative parts of the plant. For example, we can say the plant parts, root tip, stem tip, right? And another features we can say segregation and combination don't occur in this process, right? Segregation and combination don't occur in this process. So these are some specific features of mitosis, right? So we will move into the phases. So here in the mitotic stage, there are 4 phases: prophase, metaphase, anaphase, and telophase. You can simply remember this order like this. PMAT, right? So pro, meta, ana, telo, right? Yeah, here. See here that blue color area, that blue color structure, that is genetic materials, right? So just now think it as genetic materials. So prophase, see here the genetic materials are contents, right? Here the genetic materials are contents in the prophase. In the metaphase, somewhat it arranged in some way. And see here in the anaphase, it's somewhat moving into the poles. And in the telophase, you can observe into different cells. So this is the microscopic image of these mitotic stages, right? So first one is prophase, right? So prophase immediately follows S phase and G2 phase. That means it's immediately follow the G2 phase of the cell cycle and is marked by the condensation of the genetic material. So we already told here. See, condensation of genetic material, right? So here that in the prophase, that condensation of genetic material to form compact mitotic chromosomes. It is composed of 2 chromatids attached at the centromere. So now you know what is centrosome, centromere, chromatin, chromosome, centrioles, those things, right? Please be clear. Now what is going to happen here? That chromosomes, that genetic material going to condensate to form a chromosomes with 2 chromatids, Right? The completion of prophase is characterized by the initiation of the assembly of the mitotic spindle. Right? We will discuss one by one. Altogether, I will discuss about the prophase, and we will check one by one how the process is happening. Right? So we can say in the prophase that early prophase and late prophase. Right? In the early prophase, that chromatin in nucleus contains to form visible chromosome. Right? So in the G2 phase, the chromatins are like bundles, right? So now in the early prophase is contains to form a visible chromosome with two chromatids, that means sister chromatids, right? So mitotic spindle, those will forms from fibers in the cytoskeleton or centrioles. Right? Here you can observe the microscopic image, the nucleus, cytoplasm, and nuclear membrane and chromosome. In the early prophase, somewhat you can observe the nuclear membrane, but in late prophase, you can't observe the nuclear membrane. That in the late prophase, the nuclear membrane and nucleus are broken down, and nuclear membrane will disappear. See here, it started to disappear. See this nuclear envelope, nuclear membrane started to disappear, and the chromosomes continue to condensing, and now they are clearly visible. See here, they are condensed as chromosomes. The chromatins are condensed as a chromosome, and it's visible now, right? And another thing, fibers. The spindle fibers we already discussed in the early discussion of the cell division. What is the spindle? It's a like fibers, right? So spindle fibers hold kinetophores, right? Kinetophores. Right? This one, kinetophore attached to the centromere of each chromosomes. We know centromere. Already we have discussed what is centromere. What is kinetophore? The kinetochore is a complex protein containing structure to which microtubules attach. Right? So if it is chromosome, so this area is centromere. Right here, there are some structure. This structure. I couldn't able to make round here. Here, from this structure, that spindle will be formed. That because it is a complex protein containing structure which microtubules attach. So from these microtubules, that spindle fibers will form. Right. So this is that known as kinetophore. Right. So now you can observe here that how centromeres are appearing, and in this G2 phase, you can observe these centrosomes, how they appeared and how that position. Now you can see here, see earlier it's together, by side it's together, that centrosome. Now it started to moving. right? So these processes also happening here, right? So this is the spindle forming. So how the spindle form? See, these are chromosomes arranged in a plate, and this area, yeah, this area is kinetochore. From this kinetochore, spindle will be, right, and these are the centrioles. Here the centrioles, and this is that means mitotic center, right? And now one more thing is aster. What is aster? So here, until now we didn't discuss about this name, no? So now here you can observe the aster. See, this is aster. This is aster. Right? The aster is a cellular structure. It's shaped like star. It is consisting of a chromosome. It's associated microtubules during the early stages of the mitosis. So we can say it's happen in an animal cells, and also please remember that it's do not form during the mitosis in plants, right? So it's in animal in the early stage. In plant, it will not form, right? So what is that? Aster is a cellular structure shape. It's like a star. It's consisting of centrosome and associated microtubules. So you can observe in which stages? In the early stages of the mitosis. And these astral rays, these fiber rays, it's composed of microtubules. It's radiate from the centrosphere, and it's look like a cloud. See, it's look like a cloud. And what is the purpose here? What is the advantage, role of aster here? So we can say aster divides the chromosomes to ensure that each daughter cell tries to write complement chromosomes, and it also helped to and position the spindle apparatus during the so this aster also helped the spindle organization and the positioning, right? And another thing, the role of aster is it also determines the site of cleavage. Determine the site of cleavage, right? CLEAVAG, right? Cleavage furrow that splits during the cell in half during the cytokinesis, right? Cleavage furrow will be formed during the cytokinesis to splitting. That means to form two daughter cells, right? And we will move into next phase. So are you clear with the prophase? Prophase, are you clear with the prophase? Yes or no?
Yes, ma'am.
Yeah, we will move into metaphase. Right? So in the metaphase, the chromosomes to the kinetochore fibers, and it's moved to the center of the cell. Right? Chromosomes are now lined up at the equator. Right? So here, at this stage, the microtubules starts pulling the chromosomes with equal force, right? So already it's started to pulling, right? And the chromosome ends up in the middle of the cell. Right now, it ends up and lined up in the middle of the cell. Right. So this region we are talking that middle of the cell, cell. So this middle of the cell or the center of the cell, that region, this region is known as the metaphase plate. Metaphase plate. Right. So that each cell gets an entire functioning genome. Right. So here you can observe that chromosomes are up at the equator, which is known as metaphase plate, and now we have centrioles and asters. The chromosomes are connected with spindle fibers, and it's arranged and lined up in the metaphase plate. Right. So this is the microscopic image photograph of metaphase. How it's, see, you can observe in the real microscopic image. These are arranged in the metaphase plate. The chromosomes. See the asters at the poles, how it looks like through the microscope. So spindle fibers. So after you came here, if we have time to see these things under the microscope, we will check this one, right? So at least one stage you can observe under the microscope. So now you know how the prophase will be appear under the microscope, how will be the metaphase will be appear under the microscope. See here how clearly it's arranged. Right? So, and remember, when you drawing these things, you should draw each and every faces in your own, right? So, if you are drawing this metaphase, how will you draw? Cell, and then okay. You need to draw the centrosome perpendicularly. Okay, right. So from that, it's arranged. Right. Then spindles come in, and it's connected with spindles are connected with kinetochore. So those things you should remember. So all these drawings have some key points for your markings, for your grades. So if I ask the questions, diagrammatically illustrate the phases of mitosis, then what is the meaning of diagrammatically illustrate? You need to draw and mark the particular structure, parts, and you need to give some key points. That's enough diagrammatically illustrating, right? For each and every phases, you need to draw and familiar with that. Okay. Next one, the anaphase. So anaphase, it occurs rapidly, and the sister chromatids are pulled apart to opposite poles. Right. So here the sister chromatids are pulled apart. So these things you all know, you have studied in the A level. But the thing is, you should remember the keywords and terminologies. So if you forget the terminology, you can't write the exams, right? So that you need to tell that in the anaphase, so there are sister chromatids arranged in the plates in the metaphase. Now here it's going to pull apart. Already in the metaphase, the chromosomes are arranged. Now in the anaphase, So in the chromosomes, there are two sister chromatids. So they are going to pull apart to the opposite pole. So here in this one is one pole, this another pole. So these are the opposite poles. So these sister chromatids will be pulled to opposite poles of the cell kinetochore fibers. So we already told every chromatids are connected to the kinetochore fibers. So with the help of the kinetochore fibers, it will pull toward the opposite. This is pulled here, this is pulled here, and this is pulled to opposite. So now they are separated. Right? So the splitting of the sister chromatids marks the onset of anaphase. So here that key characteristic is splitting of the sister chromatids. So that is the mark of the onset of anaphase. So these sister chromatids become the chromosomes of the daughter nuclei. Right? Understood? So now these sister chromatids are separated and moving. Then later, these sister chromatids will be the chromosome for the new daughter cells. Right? So the chromosomes are then pulled towards the pole by the fiber attached to the kinetochore of each chromosomes. Right? So the centromere of each chromosomes lead at the edge while the arms trail behind it. Right? So this is happening here. So you need to here with drawing this one, and also after drawing you need to mark this. What is this? And you need to mark this one sister chromatids. This pole, and these are the spindle and kinetochore. You should mark these things when you are go for the exam writing. Right, see here, this is the real microscopic picture. See here how it appears under the microscope. See, the sister chromatids are being separated. That means not fully separated, it's being separated. Right? Okay, next one. Telophase. So in the telophase, here the sister chromatids at opposite poles. So now the sister chromatids are fully separated. Right? So the spindle resembles and nuclear envelope forms around each set of sister chromatids, and nucleus reappears, and then cytokinesis will occur, and chromosomes reappear as chromatin. Right? So this is the stage. The chromosomes that cluster at the two poles start coalescing into an undifferentiated mass. So it look like in the start. In the start, it's like a complex mass of the chromatin, isn't it? Then it's content to form chromosome. So now the process is reversed. That it's happening reversely, right? So now the sister chromatids are moved to the opposite poles. Spindles will disappear, and it's going to separate. Now these sister chromatids are coming to opposite poles. Then these sister chromatids are forming as a chromatin. It's forming chromatin coils, and then after chromatin coils on the opposite side, nuclear envelope is started to form. It started to form around the sister chromatids. After the sister chromatid come to the opposite pole only, it will happen. And now, after forming of the nuclear envelopes, the nucleus is reappears. See, nucleus is reappears. In which stage the nuclear is disappears? In which stage nucleus is disappeared? Tell me quickly. You people are there? Yes or no?
Late prophets. Prophets.
Profess? Is that? The profess is M face, no? Please clear my idea. That person said nuclear envelope disappear in profess. Is right or wrong? Sometimes I may confuse you. Samina, is that right or wrong? Nihara,
I'm wrong. Synthesis space disappear.
is that? Then you people are not clear. And who said nuclear envelope disappear in the profess? Who's that?
Ma'am, me.
Navodi.
Yes, ma'am.
Yeah. So why you are telling that? How you confident on that answer?
In that case, in the later prophets, continued condensing.
Yeah. You are right. I just want to check you are clear or not. Just you can't say it's in profess, just you can't say it's happening in G2 or G1. If you saying one answer, right, you should be clear. Right? So you can say that one person said in G2 phase. Why it can't happen in G2 phase? Because G2 phase is the growth phase, still the growth stages are happening there, isn't it? You people are clear or not? Then I need to move to that slide. Yeah. This one is G1 phase. So in the G1 phase, the cell grows in size, first growth stage. And in the G2 phase, synthesis will be happen, DNA replication, sorry, S1, S phase, the synthesis stage, that DNA replication will be happen. And next one is G2 phase. Here, second growth stage, that all the structures which is needed for cell division are made and cell organelles and proteins are synthesized. So these things happen in G2 phase. So here the thing is still the cell in the growth phase. Still the cell in growth phase. So that time cell nuclear membrane there. So that the mean is still the growth steps are happening. So still the nuclear envelope will be there. It started for the prophase, right? See, when it started for the prophase, in the prophase, early and late, in the early, that chromatin fibers, that chromatin will be condensed, right? So it started to form the chromosome. Then in the late phase, after it forming the chromosome, where the chromosomes are forming, in the late phase, that finishing the condensation of chromosomes. So at this time, this nuclear envelope will be disappear. Clear? Please let me know you are clear on this topic or not. Okay. Hansika Sandamini. Hansika Sandamini.
Yes, madam.
Nuclear, where that nuclear envelope disappear and envelope is reappear, how it's happening. Clear?
Yes, madam.
Okay. Right? And next one is the cytokinesis. So here you can observe two daughter nuclei are formed at each pole, and here you can see separate chromosomes, centrosomes, and that nuclear reappears and nucleus reappears, right? And you can see that uncoiled sister chromatids, right? The telophase. So you need to remember which phase having which steps. So comparison of anaphase and telophase, right? So here in the anaphase, it's being separated. Its sister chromatids are being separated. Here, fully separated. That means kinetophore microtubules shorten, separating chromosomes to opposite pole. That means that chromatids are being separated. And the polar microtubules along preparing cell for the cytokinesis. Right? In the telophase, so here chromosome reach pole of the cell. So it's fully separated. Disappear. Here you can't see the kinetophore because it's and another thing, the polar microtubule continue to elongate, preparing for the cell for cytokinesis. So nucleus reforms, nucleus reappears, and chromosomes decondense. That means it's uncoiled. Right. Next one is final one is cytokinesis. It means division of the cell cytoplasm and division of the cell into two halves that called daughter cells. And in plant cells, the cell plate form at the equator to divide cells. In the animal cells, that cell plate is known as cleavage furrow. So here cell plate will form. Here cleavage will form to split the cell. Right? So you can see what is cleavage furrow and what is cell plate. So this is that. So this is, see here, this is in animal cell. So this is cleavage furrow, right? And in plant cells, that is called cell plate. So you can see in one animation, you can see that mitotic stages. See how it's happening. So now in anaphase, it started to moving opposite. That sister chromatids are pulling on opposite side. Then finally telophase. So metaphase, that chromosomes are arranged in a metaphase plate. Now in anaphase, it's moving, migrate toward the poles, and telophase finished. Right? So cytokinesis completed the cell division, the cell cycle. So this is the simple animation for the mitotic stages. And I told in the beginning, uncontrolled mitosis. If mitosis is not controlled, unlimited cell division occurs, which is causing cancerous tumors. Right? So that oncogenes you can study later in the agricultural biotechnology. Biotechnology, oncogene. You can go and search that oncogenes are special proteins that increase the chance that a normal cell develops into a tumor. Right? So the oncogenes are the responsible for tumor. Right? So this is cancer cells. So that's all for the mitosis, right? So you people are clear on this topic?
Recording stopped.
You people are clear? Have any questions? Do you have lectures on 11:15? You have lectures in 11:15? Can you hear me?
Yes, ma'am. We have maths at 11:30.
11:30.
Yes, ma'am.
Okay, then quickly we can have a summary, no? Right, but what are things we have discussed today? Please respond. You people are clear or not? I need to know that we are cleared on this topic or not. Because I can't see in person. So I need to know whether it's clear. Otherwise, I need to take steps for more improving that explanation. Is that okay? I need to improve the explanation. Am I fast or you understood or not? Please let people are silent. Then the silence mean one thing. One is fully understood. Other one is you didn't understand well. That's the thing. Two meaning. With the he. With the he.
Yes, ma'am.
Are you clear in this topic, mitosis?
Yes, ma'am.
Sure?
Yeah.
Okay. Mayuri Dissanayaka.
Yes, ma'am.
Could you please tell me what are the things we have discussed today?
Cell cycle. Cell cycle in three major phases. Interphase, interphase mitosis and cytokinesis. Interphase three major steps. G1 and synthesis phase and G2 secondary growth phase. G1 cell mature and synthesis phase is DNA copy. G2 phase is prepare the cells prepare for division.
Okay, fine, good. Shashini Samaratunga. Shashini Samaratunga.
Yes, madam.
Yeah, could you please give summary? What we have discussed and the mitosis, the phases of mitosis. M phase. M phase. Stages of M phase. Are there any stages under the M phase? Then you are not clear, no? Vidusha Gunasri. Dania.
Yes, ma'am.
Yeah, can you please summarize what we have discussed under the mitosis stages? What are the stages?
Under the mitosis stage, there are two cell division, vegetative cell division and the reproductive cell division. In the mitosis stages, there are four stages. P-met, P is pro-meta and telophase. In the prophase, we can see the genetic material that contains the genetic materials, and that immediately follow G2 phase and marked by the condensation of genetic material to form the chromosomes with the two chromatids. In the prophase, we can see that one in the early prophase and the late prophase. In the early prophase, that chromatin condense to form visible chromosomes, and that will form.
Fine. Sivasamalai Vibulanandan.
Yes, madam.
Yeah. Could you please summarize the remaining part of that Dania explained?
So the next is metaphase. So here the kinetophores fibers will move the chromosomes to the center part of the cell, which is also known as metaphase plate. Then equal force will be from each opposite sides. Then it is the anaphase where the sister chromatids will be started to separate by opposite poles by the kinetochore fibers being pulled to opposite sides.
Yeah, good, fine. Satgunalingam Premnath. I just picked the names randomly. Satgunalingam Premnath. Not available. Ganesalingam Nirojan. Venurika Sandamali. No. Okay, then I can check who are those in the observation and who are those just switch on and go. Okay, so remaining telophase and cytokinesis. So this all is about you want to go for next class and you need to take break, no? Okay, so today's class, please give your comments and opinion in the chat box. Whether you have understood or not, what are the things I need to explain more, what are the things concentrate more for your facilitating your understand. Right? Okay, thank you so much. I need one more class to discuss the meiosis. So that class I will talk with your rep and we will get. Otherwise, can you please tell me the time? Yeah. Thank you, Danaska. Thank you, Naudi. Yeah, Henry, Danya. Yeah, so these things you're already familiar with that in the A level, but you need to remember that keywords, key points, terminologies. Then it's easy, and please familiar to grow and making drawing of these faces and points, and you need to label it. Labeling means you need to draw the things and you need to label. Yeah, this is chromosome, this is centromere, centriole, those things labeling. So you need to draw and you need to familiar to label. Right? Okay, thank you so much. Have a great day. We will have next class next week. Otherwise, we'll discuss. I need one more hour to discuss the meiosis. Right? Definitely you will have questions for the final from this cell division. Please remember that. Okay, thank you so much.
Thank you.
Thank you.
Thank you.
Thank you, madam.
Thank you, madam.
Thank you, ma'am.
Thank you, ma'am.
Thank you, ma'am.
Himaru, are you there? You have asked question. Himaru, are you there?
Yes, ma'am.
Yeah, that you are asking the definition of cell cycle.
Yes, ma'am.
Yeah, that means what is cell cycle? Tell me. What do you have idea for cell cycle? Just remember, because don't mug up the definition first. I need to know what in your mind regarding the cell cycle. So we can say there are growth stages, synthesis stages, again growth stages and some other divisional stages. Those are happening, no, in the cell cycle, isn't it?
Yes.
Yeah. So you need to know the process first. Now we will define the term cell cycle. So we can say the sequence of growth and division of a cell. Simply we can say that sequence. Sequence means
Pattern.
right. Sequence. I'll type here. Sequence. Sequence means one by one, step by step. There are some processes happening, no? So we can collectively say the cell cycle is the sequence of growth and division of a cell, and it consists of three major phases: mitosis, cytokinesis. This is enough for that definition for cell cycle. Clear?
Thank you.
Yeah. First, you need to know the process, then you can develop the definition. If you mark up or if you remember the definition by saying one by one, you will forget during the exams. Right?
Yes.
To understand the process, then you can develop the definition with the key points. Okay?
Okay, ma'am.
Okay, okay.
Thank you.
Excuse me, ma'am. Excuse me, ma'am.
Yes, yes.
Ma'am, my connection error. You called my name that time.
Yeah.
Sorry, ma'am.
Now can you answer for my question?
Yes, ma'am.
Yeah, so how can you differentiate the anaphase and telophase? So when I ask this question, that image should be coming to your mind, right? That one side anaphase,
Yes.
telophase. Then quickly say what is the difference, main difference.
Anaphase, ma'am, occurs rapidly. Sister chromates are pulled apart to opposite poles of the cells by kinetotic fibers.
You are reading?
Yes, ma'am.
Okay, the thing is, you should remember that anaphase, that means it's that chromatids, sister chromatids are started to opposite sides, right? That being separated. But in the telophase, you can say it's separated. Fully separated. So here being separated, here fully separated. Yeah, that's all. Then things you can add with your answers. Main, I asked main difference, right? Okay.
Okay, ma'am.
Okay, thank you.
Okay, thank you, ma'am.
Excuse me, ma'am.
Yes, tell me.
Today, which time I joined quiz?
Today we can't have. So there's some time to reschedule that. So we can't reschedule without asking the coordinator, right? So when we reschedule, that time you need to write.
Okay, ma'am.
So that one or two students who are those joined newly, they also didn't write the exam. So we gave excuse to them because the last week they have joined, right? So we will conduct one special quiz exam for them. That time you can join.
Okay.
Okay.
Thank you.
Okay, bye.